Before we get into like the platform and the technology, I want to just start off with the science for the audience who may not be as familiar with just what the nature is and importance is of, for example, modeling the way that proteins, for example, fold. You had a quote on your blog. You said, rational drug design is the idea that we can engineer medicines by understanding and then deliberately manipulating how biological machines fit together. So. And a lot of that seems to be pointed specifically at proteins, but maybe there's more beyond that as well that you can share. But maybe starting with proteins, like why are proteins important and why is it useful to predict their structures?
So basically, you can think about proteins as sort of like being the machinery that biology uses to do a lot of the functions that our body have to operate to operate this living society that our bodies and nature is. And so now the way that these machines work is sort of like these interactions that they have with one another. So every machine, every protein has a particular shape, and their function is given by how they interact with other machines, how maybe they come together, different proteins to form a bigger machine, and then how that interacts with, for example, the DNA, the RNA, and other molecules and other proteins that we have in our body. And so, understanding kind of the structure is a bit about, can start telling you about, kind of what a machine can do. It's a bit about like looking at, an object in isolation in the world. And to some extent, looking at an object in isolation is not necessarily telling you their function. You kind of really want to see it in operation. And so, the way that we can start, thinking about, seeing kind of the object in operation is trying to understand the interactions that, those proteins have with other proteins, other molecules in our body. And so, understanding interactions is really the way that we understand a lot of, molecular biology, the way that we map, often in biology study all this mechanism of actions, and typically the mechanism is, proteins has an effect, protein A has an effect on protein B has an effect on protein C, and so on, this cascade of operations. And so these are all interactions. And then, furthermore, if we want to intervene on kind of biology, for example, to make new therapeutics, what we do is that we engineer proteins or small molecules that go and interact with this protein in a particular way to change or adjust their function. And so understanding kind of these machines and how they work and how you can alter their function is critical to rationally design medicine. Now, why do I say rationally? Because the way that we used to discover medicines is just by if you want by chance or by brute forcing, trying to look for things or trying to get nature to brute force for us. And so that's kind of a bit of a new approach, and arguably kind of the approach that we need if we really want to tackle a lot of the unmet kind of needs in human disease.
Great. So you mentioned human disease there at the end. I'm curious, if there is an illness, whether it's let's say Alzheimer's or diabetes or any of the other illnesses that plague humanity. How do we have a sense of which proteins we need to be focused on and looking at that relate to that specific disease?
That's an incredibly tough problem. It's the problem of target identification. This is, I would say, still probably the toughest problem, in the whole kind of cycle of creating a drug. Also because it's the one that is probably less automatable. It really comes down to understanding biology, and typically that really stems from a lot of basic research. And so even like you mentioned Alzheimer's, I don't know that we know the best targets for Alzheimer's and all the kind of understanding. And so the other challenge is sort of like once you identify a potential target, you say, okay, maybe this protein is linked to or this particular function of this protein is linked to this particular disease. I'm going to go and create a drug for it. Now how do you know that you're right? And what you can do is you can now with modern technology you can try to knock out that gene. In some cases you can do some ways that you can sort of like validate that target, but then really you have to create a medicine for that target and then test it, and then you would test it in a mouse, see that it works in a mouse or some other animal. Now animals are not humans, and so in practice we still see often, especially in phase 2 clinical trials, many failures due to efficacy, and that's really often a problem of not that maybe being the wrong target in the first place. And so finding the right target is an incredibly challenging problem.
Great. So once we found the right target, understanding the proteins that interact with that target in a way that is meaningful, that's scientifically helpful, is really useful. And so your platform helps us do that. You mentioned molecules as being this thing that binds with a protein. So we hear of small molecules, which seem to be like little pieces of basically inert drugs that we might take as a pill. There's also antibodies and other types of proteins, small proteins that we might be able to have that bind against those. So could you share a little bit more about, so there's the protein that we've identified, understanding its shape is important, but then what are the kinds of things that are binding to it that are meaningful?
Yeah. So as you were saying, we have this target. We want to do something to this target. Often it's inhibit that target. Sometimes is activate that target. But we want to do something to that target. Now there are many kinds of therapeutics. I think I won't go. For simplicity, I will kind of stay on kind of what are called small molecule and biologics. Kind of outside of that category, there is cell, there's vaccines and kind of all sorts of other therapies. But staying between small molecule and biologics, this is really I'm creating either a molecule or I'm creating a protein that goes and interact with that target and does what we want it to do. And so within that, goes and interact with that target that, is already, like a challenge. We need to get something that goes in the right place, whether, that's the right part of, inside of the cell, or that's the maybe tumor environment if we're talking about cancer. So that's one challenge. But then, the second challenge is once you're there, can you have the right kind of type of interaction with that target? And then we also need a bunch of other things, for example, toxicity. You shouldn't go and mess up a bunch of other things, and clearance. Once you have the molecule in the body, can your body break down the molecule eventually, and so on, and things like that. And so basically, what you have to do, once you define kind of the target, is really define kind of all the properties that you would want from the molecule that you're trying to make, the molecule or the biologic, and that forms what people call the target product profile. And so that's sort of like a shopping list for what you would like, and then you would use something like our platform and so on, all sorts of complex interactions and iterations to find a molecules that fits all those desiderata. That is your profile.
And just for folks who haven't seen the way in which proteins are shaped, I would maybe inform the audience that they are these incredibly intricate, almost ribbon-like structures. And so just the thought of something being able to fit into that shape to begin with is mind-boggling, but then being able to fit in the right way to then activate the protein in a specific way, I think is even more fascinating.
Yeah, no, I mean, this is really what attracted me to structural biology in the first place. I think, just the fact that, thanks to, amazing technology that people have developed in the past 100 years, we're able to see, these minuscule, machines and, basically study kind of the structure, and you really see kind of, this, invite kind of all the audience to go and look at the shape of different proteins, and especially some of proteins like enzymes are like really kind of moving machines. Go and see kind of how yeah, they're basically miniature machines that have a super precise level of operation, and yeah, and so just you can try to predict them, and then you can visualize the results. Is something that I think is very compared to other kinds of machine learning where it's just numbers. We're able to when we make a new protein, we actually visualize it. That's very rewarding, at least for more visual people like I am.
For sure, they almost look like works of art that you could place on a desk. That's great.
Some people do.
Oh yeah.
Some people, yeah.
Dreaming of proteins, creating them,
decorating and once you make a successful one, you can 3D print it and you can put it on the desk.
Look at that. There we go. A new niche of art we've discovered. I'm curious, for a given illness that we're trying to treat, is it typically like there's this one specific protein that is interacting with it and that we need to bind to in a specific way, or is there a multitude of them? What is the general tendency of biology?
So the way that people largely think about many diseases is one target, one disease, but in practice we know that so many diseases are not single targets, and so people often still go and try to find a molecule that hits that particular target, but often then you would want to put that molecule together with maybe another one that has a similar effect, that it's complementary, and then sometimes you even stick some of this together within the same bigger molecule, or you just give them together as a mixture, and so there's this kind of ways that you can sort of like try to go about try to target all the multitudes aspect of that disease, but in many cases for example there are many, I think, examples that are known where one we didn't really understand why one particular molecule was more effective than another one, and then later on it was discovered that it was all actually because it had some secondary target interactions with another target that was also kind of critical for that disease. So in many cases, there are some, I think, targets that we just, don't really understand. But yeah, it just adds to the puzzle that, And on top of that, kind of some of the other challenge, to your point, when you're trying to make a drug, sometimes you have to be selective to a particular target, and there are maybe targets similar, or maybe. And in case of a cancer, maybe a target is mutated in the tumor, but not mutated in the wild type. And so you only have to hit, a particular variant of that target. So you have to be very selective. And in other cases, you have to be pan many of these targets. For example, sometimes in viruses, you have to be careful that they can mutate. And so if you're too selective, and the virus will be can easily escape, and so there's all sorts of different complexities that start to come into play.
Nature is a wily beast.
It's a widely dynamic, complex, and we are very far from understanding it.